Hello everybody. We are now studying a question from GATE Biotechnology 2019 question paper. It is related to the topic recombinant DNA technology. So the question is, the Bt toxin gene from Bacillus thuringiensis used to generate genetically modified crops is option A cry, option B crow, option C CDC, and option D CRE. So to understand this, let's first look into some background information. So Bacillus thuringiensis or Bt is a common soil bacterium. The genome contains genes that code for several proteins toxic to insects. So for decades, Bt has been sprayed on fields as an organic pesticide. Several major pests of crops like cotton and corn that are difficult and expensive to control with chemical insecticides are susceptible to Bt. So Bacillus thuringiensis (Bt) crops are plants that are genetically engineered or modified to contain the endospore or crystal toxins of the bacterium Bt to be resistant to certain insect pests. So in 1995, the Environmental Protection Agency (EPA) in the US approved the commercial production and distribution of the Bt crops. So the widely used commercial Bt crops are corn, cotton, potato, and tobacco. But currently, the most common Bt crops that are in use are corn and cotton. So they are genetically modified to produce the toxins produced by the bacterium so that they could have an inbuilt resistance. So Bt produce insecticidal proteins during the sporulation phase of their life cycle and aggregate to form parasporal crystals. These crystals are predominantly comprised of one or more proteins, which are the cry and the cytotoxins, also called delta endotoxins. So cry proteins are specifically toxic to the insect orders Lepidoptera, Coleoptera, Hymenoptera, Diptera, and also to nematodes. In contrast, cytotoxins are mostly found in Bt strains and are active only against the insects of the order Diptera. Also, these two toxins, they have different mechanisms of action. Crytoxins interact with specific receptors located on the host cell surface and are activated by host proteases following receptor binding. The Bt toxin then dissolves in the high pH of the insect gut and becomes activated. The toxins then attack the gut cells of the insect, punching holes in the lining. The Bt spores spill out of the gut and germinate in the insect, causing death within a couple of days. So the cytotoxins from Bt do not bind to the midgut proteins, but they interact with non-saturated membrane lipids. Once the cytotoxin binds to the lipids, it forms oligomers that insert into the membrane, thereby killing the insect. So while there are many genes encoding many of these cry and cytotoxins, Cry1AB is the one widely engineered into the genetically modified crops. So coming back to our question, the Bt toxin gene from Bacillus thuringiensis used to generate genetically modified crops is, the answer is cry. So what are these other options? So just to understand that all these other options are irrelevant. So these are basically proteins or enzymes derived or produced by bacteriophages. For example, crow here is a repressor protein of temperate bacteriophages like the lambda. They control the genetic switch that determines whether a lytic or lysogenic cycle will follow infection. It represses the synthesis from the PRM promoter, which is a promoter of the lysogenic cycle. And CDC are genes that code for proteins that regulate the cell division cycle. And CRE recombinase is a tyrosine recombinase enzyme derived from the P1 bacteriophage. And so the right answer here is cry proteins. So hope you all have an understanding of how these genetically modified plants work to have their own insecticidal properties. Thank you.